Hello students, welcome back to our channel, the Stephens Forensic Academy. So in this video, we are going to discuss our new topic, that is the methods of DNA extraction. Okay, methods of DNA extraction. So what is DNA extraction? Basically, DNA extraction, it is the process, it is the we can say process or a first attempt to isolate the DNA from its cellular components. Okay, it is the process to isolate the DNA, from its cellular components. Okay, from its cellular components. Clear? So these isolation methods are of different methods are okay, like we can have different type of methods such as solid phase extraction method, organic extraction method, differential extraction method, Kelex extraction method, and FTA card extraction method. Okay, so the first attempt to isolate the DNA from the human is done in the 1869 by a scientist that is known as Frederick Mischer. Okay, Frederick Mischer. Okay, so the first tissue, or we can say the first cell which are used are the leukocytes. Okay, leukocytes are the cells which was used for the extraction of DNA. Basically, if we study the certain, we can say basics of the DNA extraction. So there are simple steps. We first of all we have some simple steps. So first step is the cell and tissue disruption. Okay, cell and tissue disruption. Clear? So now what is this process? What is this step? Okay, so in this step we have to use some enzyme. Okay, we have to use some enzymes. For example, proteinase K. Okay, proteinase K are used for the cell and tissue disruption. Okay, so disruption basically this disruption process can also be carried out by boiling and using alkali treatment. Okay, this is the first step, to disrupt the cellular component. Okay, so secondly we can also use also used by boiling and using alkali treatment or by mechanical method. Okay, for example, if we have bone or teeth, if we have bone or teeth, okay, so we have to make it them into the powder form using the liquid nitrogen. Okay, using the liquid nitrogen. Okay, then we have to extract the DNA. Okay, with some processings. Okay, why decalcifying these tissue? Okay, we have to first decalcifying these tissues, then we can be able to extract the DNA from these type of tissues. Clear? Now if we say this is the first step, that is cell and tissue disruption is the first step. Now what is the second step? The second step is the lysis of cellular and organelle membrane. Okay, cellular membrane, basically cellular membranes. Okay, such as nuclear membrane, okay, plasma membrane. We have to lyse these all membranes. Okay, then we have to enter into the cell. Okay, first of all we have to break the plasma membrane. Okay, then we have to break the nuclear membrane. Okay, for the nuclear DNA. And if we want to get the mitochondrial DNA, first of all we have to break the plasma membrane, and then we have to break the mitochondrial membrane, then we can get the access to the DNA. Okay, it will depend upon the type of need, if we want the mitochondrial DNA or if we want the nuclear DNA. For this step we have to use different type of salts. Okay, different type of salts, and that salts are known as the choiotropic salts. Basically the function of these salts as these salts can act as a chelating agents. Okay, these salts act as a chelating agents. They can inhibit the activity of DNase enzyme. Okay, एक enzyme होता है that is the DNase enzyme. These reagents, we can say these salts can inhibit the activity of these DNA's enzyme, because these DNA enzymes can, we can say, break down the DNA. Okay, we can say this DNA's enzyme can disrupt the DNA which okay, which we are extracting from the cell. Clear? So the next step of the DNA extraction is the removal of proteins and cytoplasmic constituents from the cell. Okay, from the cell debris. Okay, the third step is the removal of proteins, removal of removal of proteins and cytoplasmic constituents. Clear? So like for this we have to like use different type of salts and different type of organic solvents. Okay, also then the fourth step is to the storage of DNA, extracted DNA. Okay, storage of extracted DNA. Okay, so these are the some like basic steps for the DNA extraction method. Okay, now we are going to discuss one by one every method. Okay, from we starting from the organic extraction method, that is also known as the PCI method. Okay, phenol chloroform isomyl alcohol method. Clear? So now we are going to discuss these methods one by one. First of all, we have a method that is the extraction with the phenol chloroform method. Okay, and also called the organic extraction method. Clear? So the first step, like as we discussed earlier, that the first step is the cell lysis and the protein digestion. Okay, first of all we have to like break down the cell and we have to digest the protein. Okay, so in this step the digestion with the proteolytic enzymes such as proteinase K and SDS. Okay, and SDS to break down the cell wall and break down the proteins and incubate at 56 degree centigrade. Okay, we have to like incubate the cell at the 56 degree centigrade. Okay, then after breaking down the, we can say, cell membrane, or after breaking down the like a cell wall. Okay, then we have to extract with the organic solvent. Okay, now the organic solvent are the three. One is the phenol. Just wait. One is the phenol. Okay, second is the chloroform. Second is the chloroform, and third is isomyl alcohol. Okay, isomyl, okay, alcohol. So they have a different function in the DNA extraction methods. Okay, so we are going to discuss the removal of proteins carried out by the extracting aqueous solution of containing DNA with a mixture of phenol chloroform isomyl alcohol in the ratio of 25 to 24 ratio 21. Okay, just remember this ratio. The phenol is of 25. Okay, chloroform is 24 and isomyl alcohol is the 1. Okay, so this is the, we can say, proportion of these all organic solvents. Clear? So phenol is used to extract the protein from the aqueous solution of phenol has a high density than the water. Okay. So why we are using the phenol? Phenol because it is has a high density than the water. Okay. So basically sometime it we have some difficulty to separate the phenol from the aqueous phase. So we have to add the chloroform. Okay, because chloroform has the higher density than the phenol. Okay, so they just form a bond between phenol and chloroform. So as a result, phenol chloroform mixture form a organic phase at the bottom of the tube and easily separated from the aqueous phase. Clear? Then when we add phenol plus chloroform, they will form the foam. Okay, they will form the foam. So to we have to use the antifogant also here. So that antifogant is the isoamyl alcohol. Okay, so this is an antifogant. It will like prevent the formation of a foam. Clear? So isomyl alcohol added to phenol and chloroform mixture to reduce the foaming. So during the partition, the DNA is solubilized in the aqueous phase, while lipids are solubilized in the organic phase, and protein located in the interface between these two. Okay, as we can see here diagram. So this is a, like we can see the test tube. Okay, so DNA will be present in the aqueous phase. Okay, the lipids will be present in the organic phase, and the interface between these two, okay, aqueous and the organic solvent, the proteins just remain the interface between these two. Okay, then we can like use this upper portion aqueous phase for our analysis in the lab. Clear? Because it contains the DNA. Okay, so after that we have to concentrate the DNA. Concentrate for concentrating DNA, we have the two type of method. One is the precipitation, second is the ultrafiltration. One is the precipitation, second is the ultrafiltration. So for that precipitation, first of all, we have for precipitation in this, the DNA is precipitated from the aqueous solution with the ethanol and salt. So ethanol deplete the hydration cell of the DNA, thus exposing the negatively charged phosphate group. Precipitation may occur if the sufficient we can say quantity of cations such as ammonium, lithium, sodium neutralize the charge on the phosphate residue of the DNA, forming the precipitate. Okay, I think you already understand this. The DNA has the negative charge. Okay, DNA has negatively charged due to the phosphate group. Okay, due to the phosphate group. Okay, so the ethanol will expose these hydration cell. Okay, and make it the hydrophobic. Okay, make it hydrophobic. Okay, means it will become a water heating substance. Okay, then by adding the some optimum concentration of salt, we can precipitate the DNA, and it will easy for us to extract the DNA from that aqueous phase. Clear? So for the second, that is the ultrafiltration. Ultrafiltration includes the, we can say, ultracentrifugation methods. Okay, we have some two machines. We can say we have some two instruments. One is the microcone, second is the emicron, are the centrifugation ultrafiltration device that can concentrate the DNA samples. Clear? So these are very, we can say, selected with the nucleotide cutoff. Okay, so this is the microcone. Microcone unit can be selected with the nucleotide cutoff equal to the smaller than the DNA fragment of interest. Okay, so these are the some, we can say, are the instruments which we use in the ultrafiltration. Okay, basically the step that is the precipitation with the ethanol is the most important step. Okay, can be asked in the GE Net examination also. Clear? So this is the diagrammatic representation of the this organic phase extraction. First of all, we have a sample in a vial. Okay, then we have to add the blood stain, we have to add the SDS, DDT, EDTA and proteinase K. Okay, which will cell first step, that is cell lysis and tissue disruption. Then we have to incubate 56-degree temperature. Okay, then centrifuge, we have to add the phenol chloroform isomer alcohol. Then we have to vertex and then centrifuge and transfer the upper portion, that is the aqueous portion, to the new tube. Then we have to add the trace buffer. Then we have to concentrate the sample. Then for that we will extract the DNA as per our choice. Okay, so in this method we will get the double stranded DNA. Okay, the DNA will be double stranded. Okay, the double strand DNA will be, we get this DNA. Okay, and it will be uh, we can say more sufficient. We can say it will be efficient for the PCR and RFLP technique also. Okay, so the advantage of these methods is like that the extracted DNA is suitable for both RFLP technique and PCR typing. So double stranded DNA obtained high molecular weight DNA, which is very essential for the RFLP technique. Okay, because RFLP technique need the high molecular weight DNA. Secondly, double stranded DNA. Okay, so the disadvantage of this is that it is a time consuming method and involve the use of hazardous chemicals. And in this method we have to transfer the sample into multiple tubes. Okay, which will increase the chances of error and contamination of the sample. Clear? So this is the all about the our first method, that is organic extraction method. So second method is the Keylex extraction method. So Keylex, first of all, we have to understand what is the Keylex. Okay, Keylex is the chelating resin suspension. Okay, chelating resin suspension can be directly added to the sample. Okay, we have to add the directly to the sample. Then we have this is act like a chelating agent. Okay, it will we can say surround the all the DNA's enzyme, and it will inhibit the activity of that DNAs. Okay, so The Kelex first introduced in the 1991. Okay, so Kelex 100 is an ion exchange resin that is added to a suspension to the sample. Kelex is composed of, now see the composition of this Kelex. Kelex is the composed of styrene divinyl benzene copolymer containing a paired amino diacetate ions that act as a chelating group in a binding and polyvalent metals ions such as magnesium. Okay, so it will bind the magnesium ion of polyvalent metal ions. So the magnesium ion are drawn in and bound up by removing the magnesium from the reaction. DNA destroying nuclease enzyme are inactivated. Okay, like the activity will be like as the chelax reagent will inhibit the activity of a DNase enzyme. Okay, or we can say nuclease enzyme and DNA molecule are protected. Okay, so our DNA molecule will be protected if we inactivate the activity of DNAs. Okay, clear. So the step are very simple. In this step, first of all, we have to wash the DNA. Okay, this step involves the contaminants and inhibitors. Okay, so like see, we have some inhibitors. Okay, like we can talk about the inhibitors. Like in blood there is a inhibitor, that is the heme. Okay, so in a washing we have to, first of all, we have to inhibit the we have to remove these type of inhibitors, so that will not be interfere our DNA amplification. Okay, later on, for example, like there are some inhibitors written here. For example, heme compound found in the blood sample should remove because it inhibits the DNA amplification. Okay, so the next step is the boiling. So boiling, we can say that boiling cells are the suspended in the solution and incubator 50. Okay, in boiling process, after the washing, we have to go with the boiling step. Okay, in this we have to incubate the sample at 56-degree temperature, then where the DNA's activity are not active. Okay, now for 20 minutes this pre-boiling are softened, this is the pre-boiling step. Okay, this will soften the cell membrane, so we have to break down the cell membrane. Okay, then again we have to heat the, or we can say boil that sample at 100 degree centigrade. Okay, 100 degree centigrade पे हमको इसको क्या करना होगा, boil करना होगा. Then after that we have to break down the cell membranes. Okay, then we have to add the suspension Kelex. Okay, जहाँ पे हम हमें क्या ऐड करना होगा, we have to add the Kelex resin suspension. Okay, which will bind the डाइवेलेंट मैग्नीशियम आयन. ओके, यहाँ पे क्या करेगा, जहाँ पे डाइवेलेंट मैग्नीशियम आयन को क्या करेगा वो लाइक उसको कैच कर लेगा. ओके, उसके साथ बॉन्ड बन जाएगा उसका, एंड इट विल इनएक्टिवेट द एक्टिविटी ऑफ डीएनएज. ओके, सो द डीएनएज डिग्रेडेशन ऑफ द एक्सट्रैक्टेड डीएनए कैन बी ब्लॉक्ड बाय अप्लाइंग द कीलेक्स रेजिन. ओके, क्लियर. सो आफ्टर दैट वी हैव टू सेंट्रीफ्यूज द सैंपल. ओके, सेंट्रीफ्यूज के लिए हमें क्या करना होगा कि ब्रीफ सेंट्रीफ्यूगेशन इज परफॉर्म टू पुल द कीलेक्स रीजन. ओके, देन वी हैव टू रिमूव द कीलेक्स रीजन एंड वी जस्ट लेफ्ट विद only DNA sample. Okay, we are just here left with only DNA sample. Okay, the supernatant is used for the DNA analysis and can be added directly to the PCR amplification. Okay, now here see here one thing is that the DNA here is the denatured. Okay, DNA here is the denatured, means the double standard DNA break down into the single standard DNA. Okay, double standard DNA denature होने के बाद क्या बन जाएगा, सिंगल स्टैंडर्ड DNA बन जाएगा. क्लियर. Clear. So this is this we can say diagrammatic representation of this whole process. So first of all we have to take a vial. Okay, then we have to add the blood stain and the water. So, okay, this is the washing step. Okay, then we have to incubate. Okay, at 50-60 temperature, then we have to centrifuge, then remove the supernatant, then we have to add the 5% Kelex. Okay, after that we have to incubate at 100 degree centigrade, and after that we have to consider the DNA. Clear. ओके, सो इसके एडवांटेजेस क्या आएंगे. सो दिस मेथड इज सिंपल एंड रैपिड एंड यूज फॉर अ सिंगल ट्यूब एक्सट्रैक्शन मेथड. ओके, सो द चांस ऑफ कंटेमिनेशन इज आर वेरी लेस हेयर. So reduce the risk of contamination and sample mixture. Remove the PCR inhibitor such as heme. Okay, यहाँ पे इनहिबिटर और भी बहुत सारे होते हैं. ओके, लेकिन यहाँ पे बेसिकली हम बात करते हैं हीम ग्रुप की. And fourth is the resulting the DNA extracted from the solution is fragmented a single stranded. Okay, like here, then the single stranded DNA which is stable for the PCR based analysis only. Okay, लाइक कीलेक्स बीट मेथड में हमें कौन सा डीएनए मिलेगा, सिंगल स्ट्रैंडेड डीएनए में हमें मिलेगा। और इसमें लाइक हम ओनली पीसीआर मेथड को ही यूज कर सकते हैं, बिकॉज आरएफएलपी टेक्निक के लिए हमें क्या करना होगा, डबल स्ट्रैंडेड डीएनए चाहिए। So for that we don't like we can't use this method for the RFLP technique. Okay, so this is a more rapid than the organic extraction method. Okay, so इसके disadvantages क्या आएंगे? First of all, not effective for RFLP typing because the Kelex denature the double stranded DNA and yield single stranded DNA. Okay, allows only PCR based analysis. Okay, so these are the disadvantages of the Kelex bead method. Now, if we talk about the next method, that is the FTA card paper. Okay, FTA paper. Okay, so what is the FTA paper? First of all, 1980, the FTA card was discovered by the Lee Vaughan at the Flinders University, Australia. Okay, so it's named as Flinders Technology Agreement. Okay, that's the FTA. FTA F means Flinders Technology Agreement. Okay, so the FTA card is we know what is the FTA card. Okay, FTA card क्या होता है? FTA card basically is an absorbent cellulose based paper that contain the four chemical substance. Okay, FTA card क्या होएगा? FTA paper is this absorbent cellular based paper that contain four chemical substance. जो फोर केमिकल सब्सटेंस होते हैं वो क्या करते हैं, टू प्रोटेक्ट द डीएनए फ्रॉम द न्यूक्लियस डिहाइड्रेशन। ओके, डिहाइड्रेशन एंड प्रिजर्व द पेपर फ्रॉम बैक्टीरियल growth. Okay, so DNA is stable on the FDA card at room temperature for over the several years. Clear? So a बड़ा ही ज़्यादा simple सा इसका like process है. The steps are very simple here. First of all, we have to take the FDA card. Then we have to like put a drop on the FDA card of the blood. Okay, the cells are the cells are lysed upon the contact with the paper, and the DNA from the WBC is immobilized within the matrix of the paper. Clear? So the small punch, then we have to like take the small punch of the paper, remove the FDA card blood stain and placed into the tube for the washing. Okay, after washing, then we have to like the bound DNA can be then purified by washing it with the solvent to remove the heme. Okay, such as like washing step is a necessary like to remove the inhibitors. Okay, and other inhibitors of the PCR reaction. Then after purification of the paper, punch can be seen visually, and because the paper is washed and red color is removed within the supernatant. Okay, सो लाइक यहाँ से क्या होएगा, जो रेड कलर होगा उसका पेपर का वो रिमूव हो जाएगा, एंड द क्लीन पंच देन एडेड डायरेक्टली टू द पीसीआर रिएक्शन। ओके, सो जो हमारा क्लीन पंच रह जाएगा उसको हम डायरेक्ट ही क्या करेंगे, पीसीआर रिएक्शन में ऐड कर देंगे। ओके, सो दिस इज वेरी सिंपल, वी कैन से नाउ मोस्टली यूज्ड टेक्नोलॉजी। ओके, नाउ डेज। सो इसके एडवांटेज क्या आएंगे? सो द एडवांटेज आर लाइक दैट, द मेजर एडवांटेज ऑफ इज कंसिस्ट इज द like a result may be obtained without the quantification. Okay, here we don't need any quantification. Okay, now what is DNA quantification, ये एक अलग टॉपिक है, इसको हम अलग वीडियो में डिस्कस करेंगे, छोटा सी वीडियो बनाएंगे उसके लिए। सो द प्रोसीजर में बी ऑटोमेटेड ऑन द रोबोटिक वर्क स्टेशन। ओके, वी कैन लाइक ऑटोमेट कर सकते हैं हम इसको। द वर्क लाइक द ब्लड स्टेन पंच में बी रीयूज फॉर। ओके, जो हमारे ब्लड स्टेन पंच होंगे उसको हम दोबारा भी यूज कर सकते हैं फॉर द फर्दर डीएनए एम्प्लीफिकेशन एंड टाइपिंग टेक्निक्स। ओके, so here the DNA will be obtained is double standard DNA will be obtained here. Clear? So इसके लाइक यही है हमारा। इसके डिसएडवांटेज क्या है? सो इसके डिसएडवांटेज लाइक यहाँ पे लाइक व्हेन द इलेक्ट्रिक फील्ड इज अप्लाइड दिस पेपर कैन लाइक जंप फ्रॉम वन वेल टू अदर, सो दैट इज डिसएडवांटेज। क्योंकि लाइक हमें नहीं पता चलेगा कि वो वहां से जंप कर गया। ओके, बिटवीन द वेल्स इन द सैंपल ट्रे। ओके, एंड वी कैन लाइक प्रोसेस सम एम्प्टी वेल्स ही हम उसको प्रोसेस कर सकते हैं। ओके, जो लाइक हमारा टाइम खराब भी होगा उसमें, एंड हमारे रिजल्ट भी खराब होगा। ओके, सो इसका लाइक है हमारा सिंपल, लाइक यहाँ पे हमने एक ड्रॉप डाली उसके ब्लड की, then we have to take a punch. Okay, small like a punch हम उसको में लेंगे, then we have to wash the sample, then we have to add the PCR reagents here, then यहाँ पे कोई quantification की जरूरत नहीं है हमें, then we have to like we can directly add to the PCR amplification method. Okay, so next is the solid phase DNA extraction method. Now what are the solid phase DNA extraction methods? Basically these are based on the silica based methods. Okay, silica based methods. Okay, first of all we have to like adsorb the DNA on the silica. Okay, then we have to remove the DNA, or we have to like elute the DNA from the silica. Okay, So DNA is selectively bound to the substrate such as silica particles and then released फॉलोइंग स्ट्रिंजेंट वॉश ओके देन हमें फिर क्या करना है उसके लाइक वॉशिंग करनी होगी ओके देन दैट प्योरीफाई द बाउंड डीएनए मॉलिक्यूल away from the proteins and lipids okay और अदर सेलुलर कंपोनेंट्स मोस्ट वाइडली ओके यहाँ पे लाइक हमें हमारे पास डिफरेंट डिफरेंट मेथड्स हैं सॉलिड फेज एक्सट्रैक्शन और ऑफ डिफरेंट मेथड्स such as Poisson columns okay DNIQ and pulp filler okay these are the different methods of DNA solid phase extraction so first is the Poisson extraction okay Poisson extraction क्या होएगा लाइक इसमें so this is the silica based extraction method this method is based on the phenomenon that DNA is reversibly adsorbed okay यहाँ पे हमें क्या करना होगा we have to like reversibly adsorb the DNA on the silica that is silicon dioxide that is SiO2 okay in the presence of high concentration of chiotropic salt okay here we have to use the chiotropic salt okay हमने starting में बात की थी कि what are the chiotropic salts so here we have to use the chiotropic salts okay now what is the function of these salt so these salt can disrupt the hydrogen bonding and affect the 3D structure of the macromolecule okay तो काइट्रोपिक सोल्ट क्या करेगा कि जो प्रोटीन्स है उसकी जो 3D स्ट्रक्चर होगी उसको क्या करेगा डिसरप्ट करेगा एंड इट विल प्रिवेंट द डीएनए और मिक्सिंग ऑफ द डीएनए विद अदर मैक्रोमोलिक्यूल्स ओके एंड दिस सॉल्ट कैन बी यूज्ड टू डीनेचर द प्रोटीन्स एंड फैसिलिटेट द एडजॉर्प्शन ऑफ डीएनए टू द सिलिका ओके इसके कौन कौन सी एग्जांपल्स हैं द एग्जांपल ऑफ काइट्रोपिक सोल्ट आर लाइक एज गोइडिन hydrochloride guanidine isothiocyanate okay sodium iodide sodium perchlorate okay these are the different examples of the chiotropic salts so in the aqueous solution the hydration cell okay of the nucleic acid now see this is the very important part okay of this in the aqueous solution the hydrogen shell of the nucleic acid shield the negative charge of phosphate at the phosphodiester backbone in the nucleic acid as a result the nucleic acid are usually hydrophilic in aqueous solution okay जब हमने स्टार्टिंग में भी बात की थी कि जो हमारा न्यूक्लिक एसिड होते हैं दैट आर द हाइड्रोफिलिक इन aqueous solution but in the presence of a chiotropic salt it become hydrophobic okay in the presence of chiotropic salt these like we can say these are the nucleic acids become hydrophobic and these dehydrating effect cause the chiotropic salt allow the phosphate residue to become available for the adsorption of the silica surface okay जब वो लाइक हाइड्रोफोबिक बन जाएगा उसके बाद वो क्या होएगा लाइक इट विल बी एबल टू एब्जॉर्ब ऑन द सिलिका सरफेस क्लियर सो इसमें लाइक स्टेप कौन कौन से होंगे सबसे पहले वही होगा कि सेलुलाइसिस होगा ओके प्रोटीन डाइजेशन होगा किसके हेल्प से विद द हेल्प ऑफ प्रोटीनेज के ओके देन वी हैव टू डीएनए एडॉप्शन होगा ऑन द सिलिका सिलिका एज द स्टेशनरी फेज इन द मेम्ब्रेन कॉन्फ़िगरेशन टू विच द डीएनए इन द सेल लाइज इट बाइंड एडॉप्शन ऑफ द डीएनए टू द सिलिका अकर इन द प्रेजेंस ऑफ हाई कंसंट्रेशन ओके हाई कंसंट्रेशन ऑफ द कायोट्रोपिक सॉल्ट ओके ध्यान रखना कि जहाँ पे इफ द पीएच इज एसिडिक okay we can say acidic then the 7.5 the DNA absorption of silica is typically around 95% okay अगर ये जहाँ से pH like कुछ भी वेरी कर दे तो इतना like absorption नहीं होगा so we have to maintain the pH okay this is the very crucial step and we have to like remove the any impurities away from this samples okay the adsorbed DNA we have to then जो हमारा adsorbed DNA होगा वो क्या होएगा हमारा double standard DNA होगा clear after the adsorption we have to wash the DNA okay washing remove the chiotropic agent and other contaminants the ethanol based wash solution is used and does not remove DNA from the silica okay so to remove the silicon from the silica so we have to elute the DNA okay so elution of the fourth step is the elution of DNA the elution the adsorbed DNA can be we can say electrically okay so we can say eluted by so not electrically here eluted by the rehydration okay we have to rehydrate the samples to elute the sample with the aqueous low salt concentration this eluted DNA is sub double standard and can be used for widely in the application okay so here this is the double standard DNA so we can use it in the PCR as well as RFLP technique okay both clear so इसके advantage क्या है कि yield high quality of DNA silica membrane we can say device can also be adapted for the automation okay automation में हम कर सकते हैं इसको and can be performed with the centrifugation or vacuum manifold in a single tube okay so next जो हमारा step next जो method है इसका ही सिलिका बेस्ड पे वो है हमारा DNA IQ okay IQ means isolation and quantitation okay isolation and quantitation okay लाइक इसका जो प्रोसेस है इट विल बी लाइक सेम एज दी सिलिका बेस्ड डीएनए बाइंडिंग। ओके, उसमें लाइक सिलिका बेस्ड बाइंडिंग होगी और इसमें हम एल्यूट करेंगे, लेकिन इसमें हम क्या करेंगे, पैरामैग्नेटिक रीजन हम यूज करेंगे। ओके, हम वी विल यूज हेयर द सिलिका कोट पैरामैग्नेटिक रीजन। ओके, जो पैरामैग्नेटिक रीजन होगा वो डीएनए को क्या करेगा, अट्रैक्ट करेगा और उसको बाइंड कर लेगा, एंड आफ्टर दैट आफ्टर द बाइंडिंग वी कैन जस्ट रिमूव द डीएनए फ्रॉम द सॉल्यूशन। ओके, तो इसके एडवांटेज क्या है, कैन बी परफॉर्म्ड इन अ सिंगल ट्यूब। ओके, बाय सिंपली एडिंग और रिमूविंग इन सम सॉल्यूशंस। सेकंड, इसका जो प्रोसेस है हम लाइक इसका डायग्राम से देखेंगे, like here. So first step is the silica based. Okay, here we have the silicon dioxide. We have to like wash here the we can say adsorbed DNA. Then we have to elute the DNA. Okay, so next is the, this is the DNA IQ. Okay, that is the, we have to use the paramagnetic bead हम use करेंगे. Okay, here we will use the paramagnetic bead. Okay, so this is the bead. Okay, so the DNA will bound this, and after that with the help of magnet, okay, it will like attracted to the base of the tube. Okay, then after that we have to elute the DNA from the paramagnetic wall, or we can say paramagnetic bead of the, okay, sample. Clear? So this is the silica. Okay, this is the silica. Okay, silica को क्या करेगा, DNA ये मैग्नेट अट्रैक्ट करेगा अपनी इस तरफ। क्लियर? तो ये था हमारा प्रोसेस। जो लास्ट है हमारा, दैट इज डिफरेंशियल एक्सट्रैक्शन मेथड। ओके, दिस इज वेरी इंपॉर्टेंट मेथड। ओके, some time on a crime scene we have we will find a sample which is a mixture of both like a female and male fraction. Okay, female fraction will be epithelial fraction and male will be sperm or seminal fluid. Okay, so this is the modified version of organic method that separate epithelial and sperm cell. Okay, so this method was developed by, or we can say described by, in 1985 by Petergel and its co-worker. So this method is used to isolate the female and male fraction from the mixture. Okay, this is very most important we can say DNA extraction method. Okay, in which we can like separate the female extraction and the male extraction to each other. So इसका procedure क्या है, like first of all we have to separate the female fraction, that are the epithelial cell. After that we have to like remove the or separate the sperm cell. Clear? So here the first lead involves the preferentially breaking down the female epithelial cell with the incubation in an SDS proteinase K mixture and at 37 degree centigrade. Okay, so जो फर्स्ट स्टेप तो वो तो सब में सेम रहेगा ना। ओके, लाइक सेल लाइसिस का। ओके, देन फॉर स्पर्म वी हैव टू यूज द DDT. ओके, here is the one thing is that this is the DDT, this is the dithiotretol. Now what is the function of this? We all know that the acrosomal cap is the made up of, जो हमारी acrosomal cap होएगी, okay, in the sperm है दी डाइसल्फाइड बॉन्ड। ओके, यहाँ पे क्या होते हैं हमारे डाइसल्फाइड लिंकेज होते हैं। ओके, सो टू ब्रेक दीज डाइसल्फाइड वी हैव टू यूज दी DTT। ओके, अगर ये ब्रेक नहीं होंगे, इफ दीज बॉन्ड्स विल नॉट बी ब्रेक, सो द मेल DNA विल नॉट रिलीज टू लाइक टू द सॉल्यूशन। ओके, सो फर्स्ट ऑफ ऑल वी हैव टू ब्रेक these disulfide linkage. Okay, with the help of this reagent, that is DTT, that is thiotretol। ओके, इसके स्पेलिंग देख लेना एक बार, इसकी स्पेलिंग देखें, नहीं, डाइथायोथ्रीटोल। ओके, दिस यहाँ रहा है डाइथायोथ्रीटोल। ओके, सो दिस इज विल हेल्प टू ब्रेक डाउन द डाइसल्फाइड ब्रिज दैट मेक अप द स्पर्म न्यूक्लियर मेम्ब्रेन। Okay, so sperm nuclei are impervious to digestion without the DDT. Okay, so without the DDT the sperm nuclei are impervious. So if sometime the person is the vasectomy, okay, अगर like उसने सर्जरी कराई हो, okay, that person is the vasectomized, so or we can say azoospermic, then ये वाला जो हमारा मेथड है, this method will not be used, okay, for the we can say for the extraction of we can say for the separation of a male to the female fraction, then we will go with the bichromosome marker, specific marker के साथ हम जाएंगे। okay, because we don't have any sperm, okay, in these conditions azoospermia or vasectomy, in these condition there will be no sperm। okay, so we will go with the bichromosome specific markers। क्लियर? सो अगर हम इसका थोड़ा प्रोसेस देखें तो इसमें लाइक मिक्सचर होगा, सो वी हैव मिक्सचर ऑफ बोथ मेल एंड फीमेल। okay, then first of all we have to add the SDSA EDTA proteinase K, okay, and then we have to incubate at 37 degree centigrade। then क्या होएगा, जो फीमेल फ्रैक्शन होगी इट विल लाइक remain upside of the tube, and the male fraction, due to heavy nature, it will just remain below the tube. Okay, then we have to like transfer the supernatant to the new tube. Okay, so that will contain the female fraction. So for the extraction of DNA from the sperm, we have to add the DTT here. Okay, at 76 degrees centigrade, then we will get here male fraction, that is the male DNA from the sperm. Clear? So these are the all we can say methods of DNA extraction. Clear? अगर आपको फिर भी कुछ भी चीज़ समझ ना आई हो, प्लीज़ मुझे आप एक बार कमेंट करना, ओके, इस वीडियो के नीचे, और एक बार वीडियो को जरूर देखना, ओके। और अगर आपको हमारी वीडियो अच्छी लगती है, प्लीज़ उसको लाइक शेयर जरूर करें, ओके, लाइक करें, सब्सक्राइब करें हमारे चैनल को। आगे हम फर्दर न्यू कंटेंट लेकर आएंगे, ओके, और इससे भी अच्छा कंटेंट लेकर आएंगे इससे हम। ओके, क्लियर? लाइक अभी हम थोड़ा सा लेट चल रहे हैं, बिकॉज थोड़ा सा हमारा लाइक सिलेबस काफ़ी ज़्यादा पड़ा है, और लाइक जो हमारा मटेरियल है उसको लाइक थोड़ा सा वी कैन से रीअरेंज करना थोड़ा सा, ठीक है, दिक्कत हो जाती है। लेकिन हम अपना सिलेबस जून से पहले हम कवर करेंगे, हमारा सारा सिलेबस उसको हम कवर करेंगे। ओके, सो दिस वाज ऑल अबाउट फॉर टुडेज वीडियो। ओके, एंड फर्दर इन दिस पीडीएफ यू हैव एडवांटेज एंड लिमिटेशन ऑफ डीएनए टाइपिंग मेथड्स। ओके, लाइक एज पर द हिस्ट्री, हिस्टोरिकल बैकग्राउंड। ओके, सबसे पहले ब्लड टाइपिंग आया था, प्रोटीन प्रोफाइलिंग, ओके, RFLP एंड SLP। ओके, so these are the difference here, you can read this table, okay, also। ओके, जैसे लाइक हिस्ट्री में डेवलप हुआ था जो भी मेरी टेक्निक, तो उसके बेस पे यहाँ पे मैंने एक चार्ट इसको ड्रॉ किया हुआ है। ओके, उसका आप जरूर रीड करना। क्लियर? सो दिस वाज ऑल फॉर टुडेज वीडियो। स्टे सेफ, स्टे ब्लेस्ड। ओके, थैंक यू सो मच।